So I'm just going to introduce Stefan Williams, who's from the Australian Centre of Field Robotics at Sydney University. So he's an expert on AUVs and things like that, which I'll talk to you about. But also remote imaging of marine systems, coral reefs, and also more further south. And so hopefully a lot of these techniques and things are really interesting and hopefully will be applicable to a lot of people here for research. So Stefan's going to talk about that. There's also Oscar Pizarro, who's a postdoc in the lab as well, who's done a lot of this work, developed a lot of the tools and techniques and things. So they'll both be here for the day. Obviously we'll be able to hear a bit about all the things that can be done, and they'll also both be around afterwards till about 3:30. So if you want to have a chat about any of his things, come see them.
Thanks, Tom. So Tom, we've been talking with for a little while, and we've also had an established collaboration with AIMS, so we've been out there yesterday talking with them about some of the work we've been doing. So what I'll do is I'll show you some examples of sorts of work we do, the kinds of data products that we're able to deliver with these AUVs, and some of the work that the ongoing work we're doing on interpretation, automatic interpretation of the imagery that we collect. I'll start by introducing IMOS. I'm assuming everybody's reasonably familiar with the IMOS program. Yeah? So I don't need to go into too much detail. I'll talk particularly about the AUV facility of IMOS, show some sample data products, and then go over a few results, show you the kinds of things that the scientific output that's coming out of this work. So recent highlights of recent publications and things like that. So start with an introduction to some of the data products and then show how that's feeding into great scientific output. And then we'll talk about where some of this is leading in alternative platforms and applications. So IMOS was originally set up through the National Collaborative Research Infrastructure Scheme back in 2007. Marine science was designated one of 8 priority programs of NCRIS. To date, it's been about a $125 million program, which currently has just been extended out to 2014. There's a little bit of uncertainty going forward. We're not really sure what to put it mildly. And I'm not convinced that next week's budget will give us much more insight into what's going to happen in the future of IMOS. There's a lot of people around the place trying to make sure that this investment in infrastructure is maintained. The IMOS investment really comes through a number of facilities. So there's an array of moorings around the country. There's investment in the Argo Float program, which is a big international program with a glider facility, ships of opportunity, or remote sensing. And we run an AUV facility, which originally was set up kind of as a pilot. What can we use AUVs to do in terms of biological observation? So we did a lot of work. Some of our early work was with Tom and Jody Webster and Rob Beeman off the Surveyor along the GBR. We did a number of other deployments around the country. And then in 2010, we were asked to transition more to a sustained observing mode of operation for the AUV program. So the idea was to go select key sites around the country and to return to those to see how those marine habitats are changing, to use the AUV to help document some of that change. So the AUV itself looks like this. So it's the main AUV that we use at the moment. It's 2 hulls. Originally designed and built at the Woods Hole Oceanographic Institution, but this platform is really designed for low altitude, high resolution, benthic imaging. Apologies for the poor camera work. I'm not much of an underwater videographer. But essentially what we have is a high resolution stereo imaging system synchronized with strobes that are forward and aft. So we get two strobes that flash with every image that we capture. We have a multi-beam sonar, so we can also do bathymetric profiling while we're underway. We carry CTD, fluorometers for measuring chlorophyll turbidity, and dissolved organic matter. There's an obstacle avoidance sonar up front, so we can operate over relatively rough terrain. And then a variety of navigation instruments, which allows the vehicle to figure out where it is. So a GPS one on the surface. We have an acoustic positioning system, which the ship can relay information down to the vehicle to give it additional information so it knows where it is. It also has a DVL, which is like an inverted ADCP. So It measures velocity of the vehicle relative to the ground, but it can also measure current profiles under the vehicle, between the vehicle and the seafloor. So all of this data is collected while it's underway. Depth rating is to 800 meters, although our typical operating sort of envelope is, say, 10 to 100 meters, and we have taken it down to about 250 meters. It's the deepest we've operated down to a few canyons. With the current battery set up, we've got about 12 hours of operation before we need to recharge the battery. So we can work a full day. And typically, that will be divided up into a number of dumps. So we'll deploy the vehicle, we'll do a pre-planned mission, we'll recover it, move to another site, put it in, and then do the recharging and data transfer overnight. The data that we collect looks like this. Oh, it's very dark. That's a function of the projector, I think. Let me just see if I can brighten that up, because that will affect a few other things as well. Sorry about that. Wait, let's do this. That it's a little bit brighter.
The color?
Yeah, the color's not great. It's all right. Color, yeah. Whoa. Brilliant. It's better? All right, well, we'll try it. No. Okay, let me try one more thing. Yeah, there's a few other things that are going to not look brilliant.
Did you uncheck that automatically adjust brightness box before you changed it? It might not be on manual.
Before, not the code section.
Over here, that one.
Yeah. It looks on the computer screen, but it doesn't.
Yeah, it's adjusting it here.
But it's the C4 is the other ones, the other panel. See, that's.
Oh yeah, sorry. Ah, so I've been calibrating the wrong one. Sorry. That's a bit brighter. And this generally makes it bright as well. Hey, all right. Okay, sorry about that. So this is imagery that we collected. Still looks better on here, so if anybody's interested later. This is data that was collected at the Ebolas Islands a couple weeks ago in WA. The AUV is flying 2 meters above the sea floor. This gives us a footprint of about 1.5 meters across here. It's one of the two stereo pairs, and the images are collected at 1.5 megapixel cameras each side. We take images at 1 hertz, so one frame per second, and we're moving forward at approximately 1 knot, so half a meter per second, and you can see we get multiple images of each bit of the seafloor as we go. And one of the big focuses early on for us was in using this sort of information to build detailed seafloor models by combining all of the images that we're collecting. So here's an example of a dive we've done in Tasmania. The vehicle started out here in about 32 meters of water, swung up to the depth of about 4 meters, and then it's conducted this mow-the-lawn kind of pattern back and forth, achieving dense coverage in a 25 by 25 meter box. And this has been selected as our kind of replicate. So typically when we're doing these repeat monitoring dives, we'll put a number of these 25 by 25 meter patches out, and then the AUV will go and do that. And that gives us a little bit of flexibility. You know, the navigation's within a meter or two, depending on the satellite, GPS satellite configuration. So there's a little bit of slop there, but the vehicle is able to revisit these patches. The structure here is derived exclusively from the stereo data, and this is a texture map, three-dimensional version of that seafloor model. And these are urchin barrens that are off St. Helens in the northeast of Tasmania. So we look at that in a little bit more detail. This is part of one of those models. You can see sort of the kelp sitting up on top of the boulder field. The resolution here is on the order of a couple of centimeters. So it's pretty high resolution data. We're stitching together tens of thousands of images to achieve these sort of seafloor models. And we've done collected this sort of data at sites all around Australia. So we sat down with our key partners, many of whom seem to be focused on the temperate areas. So we work with people from CSIRO, UWA, UNSW, the University of Tasmania, UNSW as well. And they designated a number of these sites along the east and west coasts. And then the work up in the tropics has been predominantly driven by AIMS. And I think that's one thing that I'd like to see, getting a little bit more input from a center like this. And choosing the right sites. What are the kinds of places that we should be including in these annual surveys that we're doing? Each of these symbols is sized by a number of images in our archive, colored by sort of dominant habitat type, and then showing the years that we've actually conducted these surveys. And in fact, these ones we've now repeated for a 4th year running. So we've been at WA in early April, conducting the 4th year of these observations. So we're faced with a huge amount of data. Each dive consists of tens of thousands of images. We have millions of seafloor images now from all over the country. And this for us is an interesting area that's been driving a lot of our research recently, is how do we take this raw information, these raw images, and turn it into information that's actually of use for scientific study? How do we help our end users to manage this enormous volume of data? The detailed analysis is pretty laborious, so sitting down and designating what's under 50 points is a time-consuming exercise. And even making sense of what's in this data, what is there, is a difficult thing to do. So I'll show one example. This is a data set off the Tasman Peninsula. It's 11,000 stereo pairs collected over a rocky reef in the southwest of Tasmania. And in this case, we had some hand labels available. So prior to the dive, we had available this bathymetric data. So you can see where the reef was. This is about 35 meters here, going out to about 90 meters of water depth. And the objective here was to understand the distribution of habitats along this reef. So we've designated a number of, this is actually 2 dives. These are 2-kilometer long transects, and then the vehicles kind of come back up into the shallows. So we can stitch all this imagery together, and we can look, so rather than looking at individual images, what we're able to do then is to look at sort of an aggregate mesh of the entire site. So again, this is 2 kilometers long along that main axis there. And what we end up with is this 3D model of the seafloor that we've taken. So there's the mesh. This is generated from the stereo data. We're stitching together all the individual stereo pairs to get this watertight mesh, which is then texture mapped with the imagery. So we can look at where sponges are relative to each of these mounds. We can get a better understanding, I think, than looking at individual images. We can kind of pull back, look at the distribution of these habitats, and really get a good sense of what's on the seafloor. So if we go sort of further out, this is into the deeper area. You can see we're out into kind of flat, sandy areas. There's one way to explore this data. So rather than sitting there clicking through 11,000 images and trying to figure out where all these came from, we fly through these sort of three-dimensional models of the habitat. Another thing that we've done a lot of work on is actually unsupervised clustering of the data that we've collected. So here what we've done is we've taken each image and we've described it by color, texture, and in this case, structural complexity. So we actually calculate rugosity over each image and over the aggregate meshes, and I'll talk a little bit more about that in a moment. Then we put that into an algorithm that's gone in and it's looking for similar groups of images based on these features which we describe each image. And so each row here, and the colors around the borders, correspond to a group that the computer said these things are kind of similar in this feature space. And it automatically figures out what, how many clusters it should use in order to have a description of the clusters that appear in this data. What we can then do is take each image, which we've now assigned a label, and we can plot that in space because we know where each of these images came from. So you can see that the blue images tend to be out in this deep water. And these correspond to screw shell rubble. The green is kind of sandy areas. As we get up into the reef, we can see these sort of reef-dominated images. And then up in the shallow water, we start to see a lot more kelp. And all of this is done without any notion of where each image was taken from. Yeah? So you can see there's good spatial correlation in the groups that are coming out, suggesting that the groups, the groupings that are coming out based on the visual properties of the images may have some correlation with interesting things. So if I'm interested in kelp images, I don't need to look at 11,000 images, so I can just focus on this group of images here. So I can use these sorts of tools to help guide further analysis. And if we also plot this against depth, you can see that the kelp kind of disappears around 42 meters, which is, I guess, where you'd expect the photic zone in this particular area to. There's not enough light to sustain it. So I think, despite the fact that this has no notion of what's actually, the algorithms have no notion of what's in the images, you're seeing interesting patterns that are coming out of the data, from the data itself. So it's a completely data-driven method for identifying these sorts of clusters. And then we can look at the distribution of these groups within the feature space. But I think looking at this spatially sort of makes intuitive sense. So the next thing, so those, a lot of people say, it's great, but what does it mean? How do we correlate that with things that we're interested in and get down to species levels? And we've now started to work on doing the unsupervised clustering at an image, within image level. But we've also been looking at supervised techniques, where we take an image. These are examples of images from a data set, again, from Tasmania. Somebody's labeled these with CPCE, so they've taken 50 points. They've said, this is what's at each point. We've then taken the image and we've segmented it into sort of small patches that have similar color and textural properties. So if you look in here, this looks like kind of a simplified version of this, where each segment has been colored by a mean color. Yeah? And then what we've done is we've said, well, wherever we have a segment that has one of these 50 points on it, we'll learn the relationship between the properties of that patch and the label that somebody's given us. Yeah? So now we're using the annotations to learn what properties correspond to these different labels. So brown macroalgae corresponds to these kelp. This is sort of substrate, which is sandy stuff. There's a lot of something called biological matrix, which seems to be the catch-all for, we can't really figure out what's here, we'll just kind of put it in there. Yeah? And so in this case, we've got, I think, 9 high-level classes that this has been divided up to. And then what we've done is we've taken this supervised training data and trained the classifier to learn the relationship between the patches in each of these images, and then we can apply that over the whole data set. And we can look at some statistics of how reliably we're getting labels that correspond within the patches to the data that we're given for training. And now what we can do is we can take the 75 images that have actually been labeled from this data set and apply this learned classification to 7,700 images that we're taking from this particular dive. So this is the dive profile here. It's kind of a grid and the depth profiles. And then each of these panels corresponds to a different species or label that's been assigned. So this is the biological matrix, brown macroalgae. I think this is coral. There's sponges and substrate. So you can see, again, there's a strong correlation. So brown macroalgae is tending to occur in higher proportion in the shallower water. And now we're not only doing presence-absence, but we're actually estimating percent cover based on this automated within-image classification. So here, each of the dark circles is the percent cover estimated from the 50 points provided by the hand labels. Yeah? And then what we've done is we've got about 75 of those distributed over the whole dive. And then we've gone and we've applied the classifier to all of the images, and we're applying them kind of over each other. And there's good correlation between the estimates of percent cover from the automatic classification against the hand label. So this looks like something that we will be able to apply more broadly once we start getting more training data. As we start to get these expert annotations, and I'll talk about a little bit of initiative that we've got going, start collecting all this sort of data to help train these sort of classifiers to do a lot of the laborious, time-consuming analysis once we have some expert input. So one of the properties that we do use to describe these images is rugosity. So we take a stereo pair, we stitch them, or a number of stereo pairs, we stitch them together, we get a model. Traditionally, and based on my understanding, rugosity is usually measured by taking a chain and laying it out across the substrate, looking at the ratio between the unstretched length and the kind of the length that fits the environment. So we've done some validation where we've actually put a chain out. There's a chain in this model. Stretched that out, measured rugosity the traditional way, and then we've gone and we've done some virtual measurements of rugosity on these models. So we can actually put a plane through there, calculate the rugosity. We can do this at various sizes, so we can change the chain size dynamically after the fact once we've collected this data. And another thing we can do is, rather than looking at these linear measures of rugosity, we can actually do this as a function of area. Yeah? So we have the whole model, we can project the surface area down onto a plane and then look at the ratio of surface area to projected area. And what we found here, this is a +1 article that came out late last year. What we did was we looked at real chain measured rugosity versus virtual chain take rugosity. If we take the virtual chain and we kind of perturb it on our model, we get a lot of variability in the virtual measure of rugosity. And I suspect that if you did the same with your real chain and kind of moved it around, you'd get very different numbers depending on what you happen to kind of be over. Whereas if we do the same exercise with the area-based thing, so we move our area-based measure of rugosity around, we get much lower variability here. The estimate of rugosity is much more stable as we move it around within our virtual model. And so to show you an example of this, this is a dive we did at Scott Reef. This is the interface between coral and sand. And this spot was actually picked. We did a 6-kilometer long transect through the middle of the Scott Reef Lagoon, looked at that model, found a transition between coral and sand, between the reef and the sand, and then designated this dive. So the vehicle went down, completed this dive, which looks like this. This is about 75 meters here by 50 meters, so a little smaller than a football pitch. Yeah, and the vehicle's just run reciprocal 50-meter transects next to each other. This took about 2.5 hours to complete. And what we've got is now a model of, a model of the whole environment. There's 3D information there, and we can really look around at individual colonies. We can look at sorry, it's easier with the mouse. Yeah, you can look at the transition between the coral and the substrate. Yeah, and really start to get an understanding. We were talking with Sean last night about this. Trying to you could use this to really. If I gave you 25,000 images, you'd have a hard time telling, where this is in relation to other things that are around it. By stitching this all together in one big model, we can now start to make inferences about, the whole environment that we've surveyed. Right. So if I just look at that model, immediately it jumps out that there seems to be 3 distinct bits to this model. There's the reefy bit, there's kind of the interface between the two, and there's the sand. So we can take our virtual rugosity measures, and we can run them over the whole model. So now rather than looking at rugosity of one image, I can measure rugosity over an arbitrarily large patch of the seafloor, and at arbitrary resolutions. So this is a 1 by 1 meter rugosity calculation. You can also do this. This is 1 by 1, 5 by 5, 10 by 10, 20 by 20. And you can see it's smoothing out the sort of local variability. In this process, we can also measure slope quite easily. So we get a local plane fit to the substrate and aspect. So which direction is this looking? In this case, it's a relatively flat bottom. But in other cases where we have a lot more sort of rugosity, you see sort of slope and aspect are interesting as well. And using exactly the same algorithm I showed before with the clustering, we can apply that to this data set. Again, it has no notion of what's there, but we get very strong spatial correlation in clusters that fall out of that. And again, this could be used to guide your sort of analysis. So if you're interested in stuff that looks like this, well, then you look through here and you ask the computer to pull out those images from amongst the tens of thousands that are part of this model. Another thing that we've been looking at with a group at the University of Tasmania and doing some work internally ourselves is that taking the imagery and learning the relationship between the imagery that we've collected and the broad-scale bathymetric profiles that have been collected with the ship. So the beige here is bathymetry collected by a ship. The black lines are the AUV transects that we've run. They've been predominantly focused on reefs. In this case, what they've done a manual analysis of the AUV images to identify presence, absence of particular organisms. And then they've looked at, and it's probably too small to read, but looked at properties of the bathymetric substrate, so things like easting, depth, rugosity, and looking at how those correlate with different species that they've identified. So there's algae, or encrusting algae, sponges, corals, and other organisms, sponges, things like that. Yeah, and then what they do in this case is they've used a classifier to learn the relationship between presence absence. So here, black is presence, white is absence, and then what they're doing is showing probability of occurrence of, in this case, kelp over the whole bathymetric profile of this area. So we've now got high probability of occurrence in the shallower waters for this sort of thing on the reef bits over this entire survey area. And conversely, they've done the same with all of those species that I showed earlier, but sponges tend to show up in the deeper areas. So you're starting to learn the relationship between what we're seeing in the AUV imagery and the broader scale characteristics of the environment that we've surveyed. Another thing that we're pretty interested in is the dynamics of these environments. How do they change over time, over short and longer time scales? So this is a survey of an urchin barren in the northeast of Tasmania, 50 meters by about 35 meters in this case. This was done during the day. Came back 12 hours later, and we repeated that survey. And you can see all the little black dots here are the urchins, which during the day are kind of cryptic and sitting down in the crevices. And at night, on the imagery, you can see they've now come out from the crevices and they're sitting, grazing on the algae. And in fact, if you look carefully, you can see lots of sea cucumbers that are also sitting up on the rocks. And the guys who've done a lot of surveys of these areas, they do it during the day. They do population counts, and they've compared the population counts during the day with population counts on the virtual habitat. And there's a good correlation. We somewhat undersample or underestimate the urchin density using this technique. But then again, they've said they've actually never seen any sea cucumbers in this area. So we're seeing things that you might not otherwise see. And this is a sort of close-up of the urchins all sitting up on top of the rocks. And there was some interest yesterday in crown of thorns as sort of in doing this sort of thing with crown of thorns. So this is the underlying bathymetry derived from the stereo data. Now another thing that we can do, or that we've noticed, is if we're not careful about how we correct for the lighting, I'll show an example of this here. You can see this striping that's in this model. Basically, this is a function of the fact that the vehicle has where it measures its altitude is towards the back of the vehicle. The camera's in the middle. And on a sloping bottom, that means that if it's going in one direction, it's slightly higher than in the other direction, the camera. Yeah? And because the water column attenuates light quite strongly, those small changes in altitude cause fairly strong differences in the lighting, the way, and in the illumination of the scene from the strobes. So one of the things we've done recently is to say, well, we actually have a lot of structural data. Yeah? The stereo gives us good estimates of the range to the seafloor. We can model the configuration of the lights and everything. And what you see from the raw imagery is that foreground objects, if we do a sort of naive color conversion, end up being bright and red, and things that are in background are dark and blue or green because the red attenuates pretty strongly. Yeah? And this results in these sort of artifacts that I was showing earlier. Oops. Yeah? So these stripes that are coming back and forth because of slight changes in the altitude of the camera. So we've been looking at methods to actually exploit the 3D structure that we have from the stereo data to get a better lighting compensation. So effectively what we do is each image is collected by the AUV. Looks a bit like that. We then use the stereo to derive 3D structure. Yeah? And in this case, this is again a data-driven model. We're currently working on slight variants of this, but what we can do is we can plot intensity in the red, green, and blue, red, green, and blue, against range from the camera. Yeah? And you can see there's a pretty strong signal here. There's a sort of an exponential decay. We can build a model of that as a function of the camera parameters, the lighting configuration, and all of that. And then we can fit a curve to this and say, well, this curve kind of describes the attenuation of light from the scene. And then we can do a normalization to basically pull this all up so that it all looks like it's imaged at one distance. Right? So we can compensate for the fact that things that are farther end up looking darker. And if we look at, there's the raw image. That's what comes out of the camera. Although we've recently put stronger strobes on, so we do have a little bit more light typically. If we do a sort of gray world correction, which assumes that everything's on average gray, we end up with that sort of red in the foreground, blue in the background. But if we compensate for distance, you end up sort of getting a much more even illumination across that scene. Yeah, and if we apply that to the whole surface model, you end up, a lot of those artifacts, which are kind of bright spots that are induced by the things being a little bit closer to the camera, disappear. As I said, we've recently just completed the 4th year of surveys in WA from Rottnest Island in the south up to the Abrolhos Islands in the north. We see very different characteristics. So Rottnest is predominantly kelp dominated with some coral. Around the Abrolhos, we tend to see lots of coral with a little bit of kelp. So it's kind of an interesting area, I think, scientifically, because because there are questions about how these sorts of environments will change the function of changes in temperature and oceanic conditions. As I said, we're doing these 25 by 25 meter patch surveys on an annual basis. We had a paper came out early last year looking at a comparison across years and across sites. So this is the Ebrola's sites that we surveyed. These are sites at Rottnest, looking at species along here, and mean percent cover is estimated from the CPC labeled data across 2 years. We now have 4 years of data, so they're in the process of sort of working through all of that. But one interesting thing was that between 2010 and 2011, there was a sustained period of hot oceanic conditions. So there was a bit of a marine heat wave that was sustained over a long period of time, so well above the sort of long-term averages. And our surveys happened to fall here and here. Yeah. So in April of 2010 and 2011, we were there, and then in April 2012, and now again in April 2013. If we look at a few of the images from around Rottnest, 2010, the coral's looking pretty good. 2011, not so much. Some of the species, there's a bit of bleaching. This, again, I'm sure there's any number of people in this room who would be much better interpreting this, but it looks like it's coming back. I haven't actually looked at the 2013 data. We just collected it a few weeks ago. For us, this presents some interesting challenges. Now we've got surveys at over multiple years, where there are quite drastic changes in these habitats. So this is the 2010 one of these surveys we did at the Ebrola's. This is 2011. When we get strong swell and or changes in current, sometimes AUV's nav is not as good as it could be, so we've actually missed a few patches. But if you look at, a lot of these coral colonies have bleached. And what we'd like to be able to do is, we're within a meter or two when we put the vehicle back down. But what we'd like is fine scale registration between these multi-year deployments. And so we've been looking at techniques that actually look at not just the structure of these habitats, but also the colors, and we can make inferences about what bits of the models correspond to each other, even in the face of pretty substantial changes. We're using a mutual information-based registration to try and find the alignment of these things that best explains the difference in positioning. And in this case, you can see that's sort of optimal with an offset of about 0.5 meter between dives in the 2 years. That's a little bit dependent on the conditions under which we collected it, but we can actually use the data again to do this fine scale registration. So we find that optimal offset, and then what we can do is we can go into this model, draw a box around it, and immediately tell you where that box was on the subsequent year. So you can see here these little colonies have changed reasonably substantially over the course of a year. This guy's is bleached, whereas those guys haven't. You can have a look at that. Different types of coral seem to have reacted quite differently to the changes in conditions. And there are other things that we, I guess, hadn't anticipated when we started collecting this sort of data. So one of the PhD students a year ago was looking through this data and saw a lot of these perch in the data we collected. So it's fish sitting on the surface. So he's actually gone in, taken one of these dives, scrolled through 10,000 images, found lots of little fish, used the stereo data to actually measure the size and estimate the biomass of fish as a function of size. So we can take multiple measurements using the stereo data. And then he was looking at, and this is published in Fisheries Research late last year, he's looking at the correlation between the underlying substrate, high relief reef, low relief reef, sand, and sort of his estimate of the biomass of these fish. So looking at the relationships between fish and the kinds of habitats. And one thing we've been talking with Tom about is the possibility of using these rugosity measures as another index with which to start to think about where we might see these fish. And Tom actually used some of this, the automated clustering techniques to find images from one of our first surveys off the barrier reef here looking for deep water anemones and the clownfish which are associated with them. Yeah, and I'm sure Tom's in a much better position to tell you all the gory details of that. But then again, once we find those, we can then put them up against the map, know where the images are. We can start to think about where these species are being found as a function of the underlying bathymetry. We have had a bunch of requests over the years for additional sensors. Can we measure power? Can we measure other things? Can we look around? We'd love to do fish census with since we're out there on a fairly regular basis. Talk a little bit about sort of future plans, but one thing we've done, this was actually at Rottnest a couple of weeks ago. So here we've taken just a couple of GoPros. We stuck them on the front and the back of the vehicle. And the intention is to be able to send, to see, is there scientific utility in collecting this sort of data? Do we need stereo? Is a single video stream enough? I suspect not. I suspect we may need to think about stereo. But there's also a huge difference in looking forward or back. So if we look forward, these videos are basically synchronized with the cameras looking forward and out. Not seeing a lot of fish up here. Yeah, but these guys, and I'm told they follow divers around, tending, there's a big yellow thing that just cruised past. Let's go and have a look. And this we saw fairly repeatedly on a lot of these dives. Yeah, and we can collect this kind of data over kilometers. So there's potential, I think, for integrating this sort of thing into these systems. I mentioned a little bit earlier about the idea of trying to capture the analysis data and make a lot of this data available online. Through IMOS, one of the mandates of the program is that all data that we collect is freely available and should be disseminated. So we proposed this program through the Australian National Data Service and the National E-Research NECTAR, or whatever, I forget the acronym. We got some money to basically integrate not only AUV data, but also browse, towed video, ROV data into the Australian Ocean Data Network. So to start getting these data sets online and discoverable, to build interfaces so that people can search for and explore this data, but also so that they can start doing a lot of this analysis. So rather than somebody getting their CPCA and designating their own set of labels, and then when you want to compare that with somebody else's labels, they're not really compatible. What we've been working on is an annotation tool that allows us to capture this analysis data, put it into a database, and then allow it to be searchable by the whole community. So that when somebody goes in and they score an image, nobody else should hopefully ever have to look at that image. Now, there's some issues with that, but another thing that we're interested in is using this data really to train the automatic tools and have these built into the system so that a lot of this clustering classification work can be exploited. Rather than having hard disk full of data saying, hey, can you do something with my data? We'd like to build this into these online tools. So one of the first steps in this was to get together a group of people to lay out a taxonomic classification hierarchy. And again, I'd say that we're probably quite underrepresented in tropical taxonomy, and this is probably something that we should work to improve. But effectively, we've got no biota, and then I think this may animate, although I may have taken that. Yeah, sorry. If anybody's interested in all the gory details, I'm happy to circulate this. But essentially, this is a taxonomic tree that incorporates some morphology and the kinds of things that you can identify in this sort of imagery. And we'll build this into the labeling so that when somebody labels something, it's consistent across all users. So the main focus of the site will be a map-based interface where you can select where your data is from. You can make sets of imagery that you're interested in working on. You'll be able to look through thumbnails of that data, look at the individual images, see associated information. So where was this image actually taken from? What other information was collected at that same time? So with AUV, we measure depth, salinity, temperature, so a whole pile of water column parameters that we're measuring. As the analysis data starts to be available, we'll be able to show sort of class distributions on individual images or across dives. And we've just got a prototype of this working, the actual sort of analysis where you click a bunch of points, label them, and store that in the database. So all this is now kind of a working prototype, and we'll be rolling this out fairly shortly. And all the data, all the AUV data will certainly be available through that. So this is that. All that's been focused on this one platform, doing a lot of this work for IMOS. But we've come to realize after talking to a lot of different people that a lot of these technologies are applicable in other areas. So we've got a number of different systems that exploit the sort of core stereo imaging and image analysis work that we've been doing. So we've got a diver-held system, small AUVs, we've got some deep water vehicles, and we've also recently been doing some low-cost aerial imaging. So this diver-held stereo imaging system is based on the AUV system. It's got a pair of stereo cameras, strobes that are synchronized with the cameras, GPS, depth, attitude, and direction. And basically, in this case, we don't have all the same information that the AUV has, but we use the imagery to figure out how the camera has actually moved. So we can do exactly the same thing, even though this is significantly less instrumented. And this has been used for some archaeological surveys, I'll show you in a minute, as well as sort of ecological monitoring. And Oscar spent a bit of time up at Lizard Island with Andrew Baird and Sean, I guess, yeah, and others, deploying this system over the reefs there. So this is what those habitats look like. So they were sort of pilot work. Go out and just try the tool out. There is a little measurement tool in here, so you can. Whoa, that's interesting. So you can start to do things like taking measurements, distances across this model. It is 3D, so you do have the sort of structure of the environment. And we can also export these models into a geotiff. So this is actually a geotiff of one of those dives that's now in a GIS. So you can start to take these layers and put them over top of other information that you might have about particular sites you're looking at. We've got a small AUV. This one's sort of torpedo-shaped. It's about 2 meters long. Stereo imaging system's in the middle here with strobes forward and aft. It also has a sonde at the front, which measures chlorophyll, turbidity, dissolved oxygen, and CT and D. And it basically has a similar navigation suite to the bigger vehicle. We've used this in a number of occasions. One of the things we've done actually offshore here, this was in the Hinchinbrook Channel, so we've actually swum the vehicle right up into the foreshore, about 2 meters of water depth. And then it swum sort of glider-like transects, basically tow-yoing through the water column, taking measurements with the sonde. And what you can see here is that you've got the estuarine water that's coming out, sort of turbid and less saline, sitting on top of a lens of the oceanic water. And so this is about a 12-kilometer long transect with the vehicle just basically bumping up and down through the water column. So we can collect things in addition to the imagery and the bathymetric data. We can collect this sort of data. And that torpedo-shaped vehicle is much more suitable for this water column work than the slower, bigger vehicle, which is really designed for near-bottom imaging. We've used that small vehicle and the diver rig on an archaeological expedition over a couple of years in Greece. So this is a Neolithic settlement site. It's about a 3,500-year-old city. These are actually the foundations of a building. This is a child's grave, a cyst grave, I think it is, yeah? That's sitting outside of the building. So we're able to, with the diver rig, just push this around and build these three-dimensional models of this environment. So this is the AUV being deployed off the beach, running at night, because the imaging conditions are a little more favorable. If you get strong sunlight, you get a lot of sort of caustics and that make it a little bit more difficult to do the imaging. Again, we get structure as well as the full texture mapped imagery. One of the things the archaeologists were doing was some excavation. We used the diver rig to document this excavation process. So they've got this sort of underwater vacuum cleaner where they've gone and they've designated what was supposed to be a square patch and haven't quite managed to make it square. But so they've gone in and they've been basically taking away layers of sediment. And we've then, over each day, documented how this process is evolving. So at this point, they pull out, this is a pithos jar. It's a jar that would have held grain or something like that in the house. So they've excavated that. And the archaeologists were pretty excited by this because they could actually see document how their excavation was proceeding. The AUV was also used to look at ancient foreshore lines. So this city is underwater, suggesting that there's been some subsidence. So that these are the old foreshore features here. So we've run the AUV in 140 meter long transects, basically running back and forth, collecting similar data to the big vehicle. And this is in sort of 1 to maybe 2 to 5 meters of water depth. So it's shallow, the vehicle can run on the surface. And sort of to give you a sense of the scale, this is the whole city as drawn in an archaeological plan. The diver rig data over 2 years was collected in this red data. And there were some complications with the Greeks running the program who were somewhat reluctant, despite assurances to the contrary before showing up, that they didn't want us to run the AUV on site. And, there was a lot of political wrangling, which meant that rather than having 2 weeks to survey the site, the AUV-based surveying had to be done in 2 days. So they surveyed the entire site in 2 days with the AUV, whereas the diver hill stuff is a little more laborious to get around. But the same sorts of techniques that we're using for the ecological work is being brought to bear here. And actually, I'll show you another example. This is some work. So all of this works also with other people's data sets. So we've been working closely with the University of Rhode Island for a number of years now. They have a program using big ROVs to do surveys of archaeological sites and methane hydrate, or hydrothermal vents. They have been in the Mediterranean. This year they're going to be working in the Caribbean. This is a Byzantine shipwreck. It's in about 300 meters of water, and they've actually used their camera system on their ROV, but they're using our imaging software to build these models of these wreck sites. So these are amphorae, which would have been the cargo of this ship that's gone down. I'm not exactly sure when this was from. We've also designed a deep water stereo imaging system. So we've got a package that's an integrated system that has cameras exactly the same as on the diver rig and the AUVs, synchronized strobes, and we've mounted this on the Woods Hole AUV Sentry on a mere manned submersible. We've done cruises. This is mostly Oscar's been leading this entirely. I guess Oscar's been leading a lot of this work. So the imaging system here is mounted on the Sentry AUV on the mere submersible, basically just strapped on and pushed around. These are surveys of methane seeps in about 1 kilometer of water off the west coast of the US. So these are bacterial mats that are associated with this methane kind of percolating up through the sea floor, and you can see some of the structure of these areas. Another thing we've done recently, one of my other postdocs has been looking at, is very low-cost imaging. So in intertidal zones in particular. I mean, this is applicable to lots of different areas, but he's basically just taken a camera, stuck it on a little rig that's mounted on a kite, walked along the foreshore, and then he's using techniques where we basically put a fiducial, so something that we know the size of. We can then take lots of single images and stitch them together into detailed three-dimensional models. We can get the scale from this thing that we know the size of. This is about a 200-meter stretch of foreshore here, where you can a 3D texture-mapped rendering, and this is zooming right into the model, getting sort of order millimeter size pixels, showing these rock pools. That's Coralina and Homicira. He's also flown this with different cameras. So one of them is just a standard RGB camera. The other one he's taken the... there you go. That's not... I don't know if that's me, actually. He's actually taken the infrared cut filter out, and then he's repeated the flight, and then he's able to stitch together these. So one effectively measurement of infrared reflectance, and then he's able to look at this normalized difference vegetation index, which looks at the ratio of infrared reflectance to green, and you can see the vegetation has a very different index to the bare rock. So you can start to see what's actually on the substrate. We got a LEAF grant funded that started this year to build our next generation of AUV. So it's going to be a single hull, similar to the Ivor, a bit bigger than the Ivor, but it'll have high-resolution stereo imaging. And we're exploring the possibility of putting higher resolution than the 1.5 megapixel. We've had a number of people say over the years, we'd really like to just be able to zoom in a little bit more. So, and of course, once we do a little bit more, it'll be a little bit more than that too, I expect. But we're working towards that. Again, similar configuration of lights forward and aft, and still maintaining the ability to have very near bottom maneuverability. So that's underway. So just to wrap up, so I think AUVs present a novel tool for collecting rich, high-resolution and georeference data. Managing the data and transforming it into data products is a key challenge that I think drives a lot of what we're doing at the moment. And we're engaging with end-user communities across a variety of different application domains. I'm hoping that there are opportunities to engage more closely with you guys here. I think there's some exciting challenges and applications that will drive a lot of these developments. Got a lot of people to acknowledge. We've got support from a lot of different people and lots of a big team back in Sydney who really keep this ticking along. And while I take questions, I'll show this little fun video again from forward-looking. These are things that we wouldn't see by this kind of. Oh yeah, there it is. Yeah. So, and the other thing, if anybody's interested, people are interested, we do have an iPad and iPhone app. So if you want to look at these models, you can have a little play. And if you click on models, there's a few different ones on there.
Yeah. A little pink phone.
I think we take questions. Yeah.
What's the cost of all this sort of thing changing over time? I mean, obviously it's a very expensive business, but is it coming down to the level where it will become more available? Like we could have our own little machine that we could drive around?
We'd like to see that, yeah. So the Ivor, I mean, we're still not talking about hundreds of dollars. The base platform that we buy was about 60 grand for that AUV, but then we've instrumented that with a bunch of extra bits and pieces. So the cost does still creep up. We've focused on buying the best cameras that we can for the job. You can get a GoPro for a couple $100 and the stereo GoPro. You can do similar sort of things, but our philosophy, I think, has been let's focus on getting the best imagery we can. There are issues with GoPros and the way they capture the data. And there are things that you could do similar kinds of things, but it's going to make your life a lot more difficult. I guess our philosophy has been let's get the Rolls Royce and see what we can do with it. And if we'd certainly be happy to explore opportunities to apply this to cheaper.
And how often do things go wrong, right? If a machine like that malfunctions down there, do you lose it? Or?
We haven't so far. Oh well, sorry, that's I lied. We did lose it once. It washed up on a beach 100 kilometers away. So we're very fortunate in that case. That was a series of I'm happy to talk a little bit about